Alright, today I'm going to be reviewing the methods of forensic drug identification using electrochemical sensors. To start with, the most commonly used illicit drug in the United States is marijuana, and impending legalization of it will only increase its use and open the door to commercially produced products that will need to be regulated and consumed responsibly. After alcohol, marijuana and cocaine are the drugs most commonly found in the blood of drivers who were involved in a crash. However, there is no portable screening method for the detection of delta-9 tetrahydrocannabinol, which is the main psychoactive molecule in marijuana, or cocaine, like that of a blood alcohol content. The classic technique for drug analysis is the forensic approach, which narrows down the possibilities until the drug is identified. The first step, or initial screening, is a visual examination. Next, presumptive and screening tests are performed to identify which drug could be possibly present. The most common test are spot or color test, which targets functional groups and diluents, which are substances used to dilute drugs and give a higher overall yield. And the response is dependent on the concentration of the drug analyte. The best known spot test for marijuana is fast blue B salt, which reacts with similar colors for some other plant materials. For cocaine, the Scott's test is normally used, which will test positive in the presence of lidocaine and procaine. Then, instrumental analysis is used to confirm the presence of the drug and determine at what concentration. The most common and accurate method is gas or liquid chromatography coupled with mass spectrometry. The major drawback the major drawback of mass spec is that the instruments are usually large and bulky and therefore cannot be used in the field. They are also expensive, require training to operate, have long detection times, and require separation before analysis. Alternatively, electrochemical techniques can be used to analyze a target analyte by detecting the current generated from an oxidative or reductive reaction. This is possible because THC and cocaine are both electroactive molecules. The THC molecule and other cannabinoids contain a phenol group that undergoes oxidation and exhibits a typical phenolic electrochemical response. Also, cocaine molecules contain a tertiary amine group that undergoes oxidation and generates a radical intermediate, which is immediately followed by a concerted proton-electron transfer. Electrochemical analysis offers significant advantages over classical techniques, such as portability, rapid detection, and low cost. The detection is also accurate and sensitive towards the target analyte while requiring small sample volumes. All of these characteristics is what makes it suitable for drug confirmation in a forensic application. To give us some background on the technique, the electrochemical technique used to analyze these samples is called voltammetry, which studies the properties of an analyte in solution or analytes that are absorbed onto an electrode while applying a potential difference inside the electrochemical cell. The device that controls the potential or the voltage difference is called a potentiostat, which has 3 electrodes inside. The cell is the combination of the electrodes, the solution, and the container that creates a charge that actually drives the reaction. The working electrode is where the voltage is applied and where the redox reaction takes place. The reference electrode has a stable and well-known electrode potential, which is independent of the electrolytes and is used as a reference point. The counter electrode completes the electrical current in the electrochemical cell. Normally, the electrodes are submerged in an electrolyte solution, which is a solution that is electrically conductive. It is made by dissolving high concentrations of a supporting electrolyte or a salt in a specific solvent, which must be inert in the potential range of the experiment. The electrolyte solution's purpose is to increase the conductivity and allow the migration of ions to balance the charge. Voltammetry actually works by using the potentiostat to apply voltage to the working electrode, creating a voltage difference between it and the reference electrode, which drives the electrochemical reaction on the working electrode surface where electrons flow into the analyte. Then, the ions in the electrolyte solution move to compensate for the charge and flow through the counter electrode, therefore closing the circuit and creating a current. The current between the working and counter electrodes is measured and plotted versus applied voltage, creating a graph called a voltagram, where the activity of the reaction is traced. The current peaks at specific applied voltages are due to specific redox reactions of an analyte, and it is what is used to identify it. What gives this technique the ability to be used for a wide range of detection is by using screen-printed electrodes, or SPEs. An SPE is a system that contains the 3 electrodes into one disposable strip, allowing for portable analysis of different analytes by placing the solution onto the sample sensing area. The electrodes are printed onto a flexible and chemically inert substrate using different kinds of inks, such as silver, platinum, or carbon, and covered with an insulating layer that keeps everything in place and only allows the sample sensing area to be the only part electrically active. They are also commercially printed for some applications which provide the low cost. An SPE can be modified using different kinds of inks to print the electrodes, or by chemically modifying the surface to increase their sensitivity or allow them to respond to different analytes. In this experiment, it was performed to test the sensitivity of screen-printed electrodes in a forensic context to confirm the presence of marijuana and cocaine in different samples, using only a portable potentiostat applying cyclic voltammetry. Cyclic voltammetry is a voltammetric method where the working electrode's potential is increased linearly with time, and after a certain potential is reached, it's ramped in the opposite direction to return it to its initial potential. And this completes one cycle that can be repeated as many times as necessary until the peaks on the graph aren't very defined. For the analysis of marijuana, the electrolyte solution was prepared by adding N,N-dimethylformamide, or DMF, and deionized water to a flask in a volumetric ratio of 9:1 to act as a solvent to the supporting electrolyte, which is tetrabutylammonium tetrafluoroborate, which was then added to the flask. A flow of nitrogen was passed through the solution to remove any electroactive oxygen that might be present, so it doesn't interfere with the results. Next, a sample of marijuana was added to the flask containing methanol and placed in an ultrasonic bath for about 20 minutes, and then was filtered in order to extract the cannabinoids from the plant material. The solution was then pre-purified in order to separate the delta-9 THC from the other cannabinoids by using thin layer chromatography. This was done by adding the solution to a silica plate with the UV indicator and used a mobile phase of N-hexane and acetone. The spot in the silica plate containing the THC was identified using a UV light and scraped off, and then it was suspended in the electrolyte solution, which was then filtered again to prepare it for analysis. Next, a commercial-grade THC standard sample in methanol was diluted to be used as another sample. This test was performed using a screen-printed carbon and counter electrode with a silver chloride reference electrode with no chemical modifications. For testing, cyclic voltammetry was performed with potentials between negative 0.5 and 0.9 volts versus the silver chloride reference electrode. First, drops of the THC standard solution in increasing concentrations was applied to the electrode and left about 15 minutes to allow for the methanol to evaporate. Then, the electrolyte solution was added. After a drop of each sample was placed onto the working electrode, a technique called pre-concentration was used, where the negative 0.5 volts was applied for about 30 seconds in order for the electroactive species to collect on the surface of the electrode. This procedure helped construct an analytical curve by plotting the anodic peak current versus THC concentration. And according to the calibration curve, there is a linear relationship between anodic peak current and THC concentration until about 20 cycles. The curve also determined the limit of detection to be 3.0 micromoles per liter and the limit of quantification to be 9.0 micromoles per liter. Also, the electrolyte solution showed an expected behavior as it did not produce any peak signals in the graphs, like in graph A, proving that it is inert in the range of the experiment. A well-shaped peak was observed after the addition of 5 microliters of the standard solution, which gave an anodic peak current at 0.18 volts versus the silver chloride reference electrode, which corresponds to the oxidation of the phenol group on the THC molecule. The previous procedure was repeated for the analysis of THC separated from the marijuana samples. The graphs from the addition of the extracted THC showed similar behavior to the standard solution, with a well-defined anodic peak at 0.16 volts versus the silver chloride reference electrode after the addition of 12 microliters, which corresponds to 3.9 micromoles per liter according to the calibration curve. The average concentration of the THC in the marijuana sample determined from cyclic voltammetry was calculated to be 3.1%. The concentration using HPLC as a reference was determined to be 3.5%. For cocaine analysis, the electrolyte solution was prepared by dissolving the supporting electrolyte, sodium perchlorate, and acetonitrile as a solvent. Cocaine solutions were made by treating a confiscated sample, which comes in the water-soluble form of cocaine chlorhydrate, with an aqueous solution of sodium bicarbonate in order to remove the hydrochloric acid. The solution was then filtered, rinsed, and dried to remove the cocaine analyte, and then dissolved in the electrolyte solution. To study interfering substances such as caffeine, procaine, and lidocaine, a sample of each was dissolved in the same electrolyte solution and then deposited on the electrode to see if there's any reactions. For cocaine, A platinum working and counter electrode was used with the silver chloride reference electrode. However, the working electrode in the sensor was chemically modified in order to increase its selectivity for cocaine using a cobalt hexocyanoferrate film. Because this was used because of its well-defined redox reaction and electrochemical response. The hexocyanoferrate film was used because they have high electron transfer capabilities, making them good electrocatalytic mediators and electrode surface modifiers. To modify the working electrode, a solution of cobalt, chloride, and potassium hexocyanoferrate was mixed in an aqueous solution of sodium perchlorate, and a drop was deposited on the surface. The formation of the film was confirmed after 10 potential cycles using a blank with just the electrolyte solution. Then, a drop of each cocaine solution in increasing concentrations was deposited on the electrode with an applied voltage between negative 0.1 and positive 0.7 volts. The graphs in graph A of the cocaine solution demonstrated the cathodic peak showed a better response proportional to the presence of cocaine. The analytical curve in graph B obtained from plotting the cathodic current versus cocaine concentrations also showed a linear relationship between current peaks and cocaine concentrations, which can be explained by the interaction between the cocaine molecules and the cobalt ions in the hexocyanoferrate film, where the drug is consumed, causing both anodic and cathodic peaks to decrease. The linearity between the peak concentrations was formed into an equation where the limit of detection was determined to be 2.88 times 10 to the negative 5 moles per liter, and the limit of quantification to be 9.62 times 10 to the negative 5 moles per liter. When looking at the interference substances in Figure C, each analyte was analyzed individually for interference in the cocaine signal by the consumption of the hexocyanoferrate film. And none of the results showed any decreasing in the peak currents, which means no change in the graphs. That suggests that the specific peak signal for cocaine enables analysis of samples just by dissolving it in acetonitrile and sodium perchlorate. When comparing the voltammetric results and the ones obtained by HPLC in Figure D, very similar results can be seen, where cyclic voltammetry determined the concentrations to be 3.51% and HPLC determined it to be 3.13%. The study concluded that the platinum screen-printed electrode with the cobalt hexocyanoferrate film surface modifier provided analysis at lower levels of concentration with comparable accuracy to classic techniques and was not interfered with by other substances that could possibly be present. The carbon screen-printed electrode and pre-purification step of THC showed good accuracy without any modification, but could only detect at levels higher than that of other techniques already used.